Assalamu alaikum, this is Snan and today we're going to talk about thyroid hormone. Thyroid hormone. Now, thyroid hormone consists of triiodothyronine, which is T3, and tetraiodothyronine, also known as thyroxine, T4. The affinity, the working of T3 is 4 times as compared to T4. Both of these hormones are produced by the thyroid follicles made up of cuboidal cells. For explanation, let us see the diagram. These are the thyroid follicles and they are made up of small cuboidal cells. And here are the blood capillaries, and the middle, the empty portion of the thyroid follicle is known as colloid. The requirement of iodine is essential for the formation of thyroid hormone. So, a typical requirement of 1 milligram per week of iodine is necessary for the formation of thyroid hormone. Now, moving toward the formation of thyroid. Now, this is the follicular cells, the cuboidal follicular cell I have formed here. So, this is the enlarged diagram of that. This is the outer surface known as the basolateral surface, and this is the inner surface, that is the colloidal surface, and we'll call it the apical surface of the follicular cell. Now, what happens? The iodine is transported inside the follicular cell via sodium iodide symporter. Symporter means it transports another substance along with iodide, and that is 2 ions of sodium. So, this iodide is then transferred to the apical surface of the follicular cell, which is the colloidal area, via a transporter called penderine. Now, penderine is a iodide chloride antiporter, means that it moves the iodide into the colloid and the chloride in the opposite direction. So, as soon as the iodide, which is negatively charged ion, enters the colloid, it is oxidized by an enzyme known as peroxidase into iodine molecule. Now, a question arises that how does the sodium iodine symporter gets the energy? It gets the energy by the sodium potassium ATPase, which moves the sodium to the outside of the cell and the potassium ion to the inside of the cell. So it typically makes a gradient of sodium. So when the sodium diffuses to the inside of the cell, it releases energy which is utilized in the inward movement of the iodide ion. Now, moving back towards the iodine. Now, this iodine is attached to a glycoprotein molecule known as thyroglobin. The thyroglobin is produced by the endoplasmic reticulum in the follicular cell, move to the Golgi apparatus, and then via a peroxidase enzyme help, the thyroglobin is coupled with an iodine to form a thyroiodine residue. If thyroglobin attaches to one iodine residue, it forms the monoiodothyronine. If it attaches with 2 iodine molecules, it forms diiodothyronine, as found here. Monoiodothyronine, diiodothyronine. Then coupling occurs. Coupling means the combining of these thyroiodine residues. When one monoiodothyronine attaches with the diiodothyronine, and the hormone triiodothyronine is formed. When 2 diiodothyronine couples, a hormone called tetraiodothyronine, also known as thyroxine, is formed. But when a diiodothyronine couples with a monoiodothyronine, means to say that if we take from the hydroxyl side, first we get the diiodothyronine and then we get the monoiodothyronine, it is the reverse of the T3, then it forms the reverse T3, which is no functional significance in human. And as I have explained earlier, thyroglobin is a glycoprotein. It is a glycoprotein and consists of 70 tyrosine residues. So contains 70 tyrosine residues which attaches to the iodine to form the tyroirosine residue, which couple and forms the T3, RT3, T4. Now after the formation of these components, the final product attaches to the pseudopod extension at the apical membrane of the follicular cell, and through the pinocytosis it moves inside the cell. Now it is inside the cell. We need the hormones, that is T3, T4, attached to the thyroglobin residues, but we don't need the thyroglobin. So inside this cell, the proteases separate the T3 and T4 from it, which then diffuse out of the cell. The amount of T3 which diffuse out of the cell is 3%, while T4 is 97%, which are transported then to the blood via the nearby blood capillaries, and in the blood they bound to the plasma protein. Now another function performed by the proteases here is that some of the thyroiodine residues do not couple to form T3, T4. So they are removed as such from the thyroglobin, that is monoiodothyronine and diiodothyronine. After this, they are deionized by an enzyme called deiodinase. So tyrosine is separated from them, and iodide molecule is iodide ion is separated from it, and this iodide is further used again in the process. This process is called recycling. Moving further, general information about the T3 is that its half-life is 1 to 2 days. It is more active, 4 times more active than T4, and the plasma protein has lesser affinity for T3. While T4, its half-life is 6 to 8 days, it's less active, and the plasma protein has higher affinity for the T4. Now the working of the thyroid hormone. Two types of working are observed in thyroid hormone, that is genomic working and non-genomic working. Now genomic working, it lasts for hours, days and weeks. The working occurs as inside the cell, the T3, T4 move inside the cell, and where T4 is converted to T3 by 5' iodinase, because T3 is more active than T4. Now this T3 binds to the thyroid hormone receptor inside the nucleus, where it regulates the gene expression, causes transcription, translation, and finally the protein synthesis. The thyroid hormone receptor inside the nucleus is a heterodimer. It contains a receptor portion and a activator or repressor portion. If an activator is attached to the thyroid hormone, it upregulates the gene expression. If a co-repressor is attached to the thyroid hormone receptor, it deregulates the gene expression. Now, with the thyroid hormone, a component of DNA known as response element is attached. The thyroid hormone activates this response element and causes the gene transcription, translation, and finally the protein synthesis. This was the genomic working of the thyroid hormone. Now the non-genomic working of the thyroid hormone. It occurs within minutes, includes examples like regulation of ion channel, for example, increased transport of sodium ion, and oxidative phosphorylation of different components inside the cell. Both of these functions causes the activation of intracellular messengers, second messengers, including the cyclic AMP and protein kinase. Both of these causes a cascade of reaction which results into several different functions of the thyroid hormone. Now, moving toward the regulation of the thyroid hormone. Regulations are two types: the upregulation, which is the positive regulation, and the downregulation, which is the negative regulation. Now, the cool temperature, the thyroid hormone increases, so the cool temperature causes an increase in the synthesis of thyroid hormone. while another upregulation occurs from the thyroid releasing hormone, thyrotroph releasing hormone, which is released from the hypothalamus. This hormone attaches to the thyrotroph cells in the anterior pituitary bypassing the hypothalamus pituitary portal system. So this attachment to the thyrotroph activates the phospholipase second messenger system, which causes the activation of protein kinase phospholipase C. This phospholipase C causes a number of reactions which causes the thyrotrophs to release thyroid stimulating hormone into the blood. Now through the blood, the thyroid stimulating hormone attaches to the receptors within the thyroid gland and causes the activation of adenyl cyclase second messenger system, which activates the cyclic AMP and causes a cascade of reaction which causes the thyroid follicles to promote every aspects of T3 T4 production. Every aspects include the increased iodine trapping, increased T3 T4 release, increased thyroglobulin synthesis, and lastly increased iodination. Now the negative regulation of the thyroid hormone include the feedback control. Feedback control means that if let's say the thyroid hormone is increased about 1.75 times in the blood, it will cause the decreased production of thyroid stimulating hormone and thyrotroph releasing hormone from the anterior pituitary and hypothalamus respectively. Now the second negative regulation is the anti-thyroid substances. It includes substances like thiocyanate, increased iodide in the blood, and propyl thiouracil. Thiocyanate causes the competitive inhibition of iodine transporter, so it decreases the iodine trapping in the follicular cell. Increased iodide production halts the functioning of thyroid hormone and decreases the blood supply to it, while the propyl thiouracil partially blocks the peroxidase which is required for the iodination in the formation of thyroid hormone and the coupling process. The third negative regulator is the excitement and anxiety. These are the emotional factors which cause an acute decrease in the TSH secretion. Now, why an acute decrease in the TSH secretion? Because in these conditions, the increase in heat and metabolism occurs, so an acute decrease in TSH secretion is performed to counter those increased heat and metabolism. Now the function of the thyroid hormone. Thyroid hormone has a vast variety of functions, including the hair follicle growth increase, increased sebaceous and sweat gland secretion, and that is caused due to the vasodilation in the skin because the temperature increases by the thyroid hormone functioning. Increase heat and metabolism. It also causes the increased heart rate and heart strength. But at excessive quantity, it causes a decrease in heart strength due to the protein catabolism. The thyroid hormone also causes the speeding up of BMR, that is the basal metabolic rate. The thyroid hormone works synergistically with the growth hormone. So it causes increase in both bone growth, increase in skeletal growth, and it also involved in the brain development. Lastly, the thyroid hormone also causes the SNS response, that is the fight or flight response, which increases the cardiac output, increases the respiratory rate, and increases the mental awareness by increasing the sensitivity of the neurons. That's all for the thyroid hormone. Now moving toward the fun facts of our episode of thyroid hormone. Do you know that BMR is increased by the thyroid hormone? So what is the reason behind the increased BMR by the thyroid hormone? One reason is that the thyroid hormone causes the increased activity of sodium potassium ATPase and increased leakiness of sodium, which causes increased energy usage by the cell for taking the sodium inside, taking sodium back inside the cell, and energy utilized by the sodium potassium ATPase. all these factors causes an increase in heat production, which ultimately increase the basal metabolic rate. Another fact, that is the effect of thyroid deficiency, are not observed for at least 2 to 3 months. Now, we should keep in mind that it is not a property of the endocrine gland to store their hormones. But why does the effect of thyroid deficiency are not observed for 2 to 3 months? The answer is the thyroid. It is an unusual ability of thyroid as an endocrine gland that thyroid hormone is stored inside its follicles for a supply of 2 to 3 months. And that's all for today. Thank you.
Yeah.